Today we say about this diarrheal diseases. Devendra. Now start with this diarrheal diseases. Before the starting, first we see about the various definitions used in this diarrheal diseases. First, the definition of diarrhea. It is increase in frequency, fluidity, and volume of bowel movement in relation to the normal habit of the individual. Now, diarrheal diseases are the one of the leading causes of illness globally, cause significant morbidity and mortality. Worldwide, there is 1.7 to 5 billion cases of diarrhea occur per year, with 1.6 million deaths, accounting for the second leading cause of death globally. Now, diarrheal diseases. First definition of diarrhea, generally I have told you. So what is diarrhea? It is increase in frequency, fluidity, and volume of bowel movement in relation to the normal habit of the individual. Now, dysentery. What is dysentery? It is passage of blood and mucus along with feces. Third term is reverse diarrhea. Then persistent diarrhea, gastroenteritis, and food poisoning. Now, other gastrointestinal infective syndromes that include acute vomiting, necrotizing enterocolitis, necrotizing enteritis, and pseudomembranous enterocolitis, tropical spore, then peptic ulcer disease, and infectious of the gastrointestinal structures such as appendicitis, diverticulitis, and typhlitis. तो डायरिया इट इज डिफाइंड पैसेज ऑफ 3 और मोर, जैसे कि मैंने बताया 3 डिफाइंड एज पैसेज ऑफ 3 और मोर लूज और लिक्विड स्टूल्स पर डे in excess than the usual habit of that the person. Then it is caused by microbial infections or as a result of other gastrointestinal diseases such as inflammatory bowel diseases, colic diseases, etc. Acute diarrhea, it usually lasts for less than 14 days and most often caused by viral agents followed by the bacterial or parasitic agents. Now, dysentery, it is characterized by diarrhea with increased blood and mucus, often associated with fever. Then there is abdominal pain and tenesmus, that is feeling of constant need to pass stools despite of an empty colon. Now, the mechanism of this diarrheal diseases. First, non-inflammatory. The non-inflammatory, the location is proximal small bowel. Illness is watery diarrhea, stool findings, there is no fecal leukocytes, then fecal lactoferrin not increased or mildly increased. The pathogens involved, first bacteria, mostly enterotoxin mediated. First is Vibrio cholerae, then Escherichia coli. It includes enteropathogenic, enterotoxigenic, and enteroaggressive E. coli. Then Clostridium perfringens, Bacillus cereus, Staphylococcus aureus, Aeromonas hydrophilia, Pleomonas shigulloides. Parasites include, first is protozoa, Giardia lamblia, Cryptosporidium parvum, Cyclospora, Cytogenesis, and Cystoisospora belli, previously called as Isospora belli, and Blastocystis hominis. Mechanism, non-inflammatory. Location, proximal small bowel. Illness, watery diarrhea. Stool findings, no fecal leukocytes and fecal lactoferrin not increased or mildly increased. The pathogens involved, ये फर्स्ट पहले आपने देखा कि बैक्टीरिया. नाउ वायरसेस. Viruses, the most important viruses are rotavirus, norovirus, calicivirus, and astrovirus. Also enteric adenovirus, serotypes 40 and 41. Fungi, it includes microsporidia. Now parasites include first helminths, Ascaris lumbricoides, hookworm, Strongyloids stercoralis, Enterobius vermicularis, Trichinella spiralis, Tinea species that include Tinea saginata and Tinea solium, then H. nana, that is Hymenopus nana, D. Phyllidium caninum, then D. Phyllobothrium latum, and Phaseolopsis buscae. Now mechanism inflammatory, that is invasion or cytotoxin. Location colon or distal small bowel, illness, dysentery or inflammatory diarrhea. Stool finding, there is fecal pus cells that includes polymorphonuclear leukocytes, they are increased, and then fecal lactoferrin, it is markedly increased. The pathogens involved predominantly dysentery, Shigella species, Campylobacter jejuni, hemorrhagic E. coli, and enteroinvasive E. coli, and also Vibrio parahemolyticus. Predominantly inflammatory diarrhea. It is caused by non-typhoidal salmonella, Yersinia enterocolitica, Listeria monocytogenes, Clostridium difficile, then Pleomonas shigelloides, and Klebsiella oxytoca. Parasite include predominantly dysentery. It includes Entomobia histolytica, Valentinium coli, Trichuris trichura, Schistosoma mansoni, and Schistosoma japonicum. Mechanism penetrating. The location, it is distal small bowel, illness, enteric fever, stool finding, fecal mononuclear leukocytes, they are increased. The pathogens involved Salmonella typhi, that is enteric fever, and Yersinia enterocolitica, which causes typhoid-like fever. Now, what is this traverse diarrhea? Most common traverse-related infectious illnesses. Clinical presentations, most cases begin with the first 3 or 5 days, characterized by sudden onset of abdominal cramp, anorexia, and watery diarrhea. The illness is generally self-limited and lasting for 1 to 5 days. And it is caused by overall enterotoxinic E. coli is the most commonest agent, followed by enteroaggressive E. coli. Also Campylobacter jejuni, most common in Asia, and norovirus associated with traveling on cruise ship. Now etiological agent or causative agent of this traverse diarrhea. First, bacteria, they are responsible for 50 to 75%. That includes enterotoxinic E. coli, commonest or single most important agent. Then enteroaggressive E. coli, it is up to 5 to 35%, emerging enteric pathogen with worldwide distribution. Third bacteria is Campylobacter jejuni, responsible for 5 to 25%, most common in Asia. Shigella and enteroinvasive E. coli, major cause of dysentery. Then non-typhoidal salmonella, commonest agent in India. Others that includes Aeromonas, Pleomonas, and Vibrio cholerae. Viral agents, viral etiological agents. Viruses, they are less than 25%. That include norovirus less than 10% and rotavirus less than 5%. Parasites up to 10%. Comments in norovirus associated with cruise ship. Rotavirus associated with common among children, and parasites that involve Giardia lamblia, Cryptosporidium parvum, Entomobia histolytica, and Cyclospora. Now the diarrhea. What is this persistent diarrhea? Diarrhea that lasts for more than 14 days, that is usually 2 to 4 weeks, it is considered to be persistent. Now chronic diarrhea usually lasts for more than 4 weeks and may result from infections due to various organisms. Parasites include Cryptosporidium, Cyclospora, Entomobia histolytica, Giardia, account for a major cause of chronic diarrhea. Bacteria includes Aeromonas, Campylobacter, Clostridium difficile, and Pleomonas. Viruses that include cytomegalovirus, commonest cause of are common in immunocompromised host. Fungi It includes Microsporidia. Again, it is common in immunocompromised host. What is gastroenteritis or infectious diarrhea? It is defined as it is an inflammation of mucous membrane of stomach and intestine resulting in combination of diarrhea, vomiting, and pain in abdomen with or without mucus or blood in stool. There is also fever or dehydration. Food poisoning. What is food poisoning? Food poisoning refers to an illness acquired through consumption of food or drink contaminated either with microorganisms or their toxins. Now its pathogenicity. Pathogenicity may first see what inoculum size, adherence, toxin production, and invasion. Now toxin production. Toxin production, first the important toxin is enterotoxin. It is produced by cholera toxins, cholera species, then Vibrio parahemolyticus and E. coli. In Escherichia coli, there is LT and ST. It is produced by enterotoxigenic E. coli, EAST by enteroaggressive E. coli, and verocytotoxin by enterohemorrhagic E. coli. Apart from that, Clostridium difficile toxin A, Aeromonas, rotavirus, and Campylobacter jejuni. Cytotoxin. Cytotoxin is produced by or responsible for Shigella dysentery type 1, enterohemorrhagic E. coli, and Clostridium difficile toxin B. Neurotoxin, it is produced by Staphylococcus aureus, that enterotoxin, then Bacillus cereus toxin, and Clostridium botulinum toxin. Adherence or attachment within or close to the mucosal cells. E. coli. Pathogenic species are enteropathogenic E. coli, enterohemorrhagic E. coli, Cryptosporidium parvum, Cyclospora species, Cystiospora species, rotavirus, and norovirus. Now, invasion of intestinal epithelium. Shigella species, enteroinvasive E. coli, Campylobacter jejuni, Yersinia enterocolitica, Pleomonas shigelloides, Enterohistolytica, and Valentinium coli. Now, suppression of normal flora, neutralization of gastric acidity, responsible factors or risk factors are suppression of normal flora, neutralization of gastric acidity, inhibition of intestinal motility, age and location, antibiotic associated, impaired host immunity, and genetic determinants. Laboratory diagnosis. Now, laboratory diagnosis can be carried out by first specimen required specimen collection. Fecal specimen containing mucal flakes collected in a sterile screw-capped white mouthed container. In carriers, rectal swab may be collected. Then specimen should be transported to lab within 1 hour of collection. And if there is likely to be delay for more than 1 hour is suspected, fecal specimens should be collected and transported to transport media. Now diagnosis can be carried out by first microscopy. See the color of specimen, its consistency, whether it may be form, semi-form, or liquid. Again, see presence of blood. If there is presence of blood, suggestive of dysentery. If mucus or pus, suggestive of inflammatory diarrhea. Then presence of adult parasites. For example, Enterobius, Ascaris, or Tinea segments. Now microscopy. Microscopy may first we have to do wet mount preparation. Here you will get whether it may be caused by bacteria or parasites. Then hanging drop preparations for suggestive of if there is suggestive of Vibrio cholerae. Gram stain smear, modified acid fast staining, generally for the parasites which are acid fast that include Cryptosporidium parvum and Isospora, commonest cause of opportunistic parasitic infection in eggs. And then lastly, electron microscopy if there is suspected to be viral diarrhea or dysentery. Then apart from microscopy, the same specimen that should be inoculated on appropriate media, cultivation or culture media, then identification of growth, then antimicrobial susceptibility test. Now tissue culture. In case of viral diarrhea. Now, tissue culture, this is carried out for the detection of enteric viruses and also for same diarrheogenic E. coli. In E. coli, diarrheogenic or enterotoxigenic E. coli penetrates HeLa and HEP2 cell line, whereas verocytotoxin and enterohemorrhagic E. coli is detected by its cytotoxic effects on vero cell line. Now, antigen detection. Antigen detection, antigen is detected by ELISA. ELISA is available for detection of rotavirus antigen in stool. Now, its rapid test, immunochromatographic test is available for simultaneous detection of antigen in 3 parasites. And these 3 parasites that include Entomobia histolytica, Diarrheal diseases, and Cryptosporidium in stool. Latex agglutination test, it is available to detect E. coli O157 antigen in stool. Rapid test available to detect Clostridium difficile antigen, that is glutamate dehydrogenase and toxin A/B in stool. Molecular methods. The most molecular methods is PCR, that is polymerase chain reaction. Assays can be carried out by for the detection of specific genes in enteric pathogens. Second molecular method, biofire film array. It is fully automated commercial nested multiplex polymerase chain reaction. Its gastrointestinal panel is used to detect common bacteria, viral and parasitic diarrheal pathogens. Other methods, methylene blue preparation and fecal lactoferrin. Now see clinical features in case of diarrhea or dysentery. If there is enteric bacteria, that includes Vibrio cholerae. Presentation, watery diarrhea. Now what is identification features? It shows darting type of motility. In gram stain, we will get comma shaped gram negative bacilli. In TCBS culture, it produces yellow colonies due to lactose fermentation. Identification is by conventional and biochemical test or by automated methods like Whitek or Maldi, TOF. Agglutinates with Vibrio cholera O1 antisera and Ujgare antisera. This is the most common pattern, though the serotypes are also present. Now, enteric bacteria Shigella. Presentation dysentery. It is gram negative bacilli, non-motile. On Meconium or deoxycholate citrate agar, it produces non-lactose fermenting colonies. On xylose lysine deoxycholate agar, it produces red colonies without black center. Identification is by conventional biochemical trace or by automated method like Whitek or Maldi TOF. It agglutinates with Shigella polyvalent antisera and specific monovalent antisera. If there is suspected as a salmonella, presentation inflammatory diarrhea. Identification features, it is gram negative bacilli, motile. On Meconium agar, it produces non-fermenting translucent colonies. On deoxycholate agar also produces non-lactose fermenting colonies with black center. On XLT, that is xylose lysine deoxycholate agar, it produces red colonies with black center. Identification is by again conventional biochemical test or by automated methods like Whitek or Maldi TOF. Again, this it agglutinates with Salmonella poly O antisera and serotype O4 specific antisera. If viral pathogens are suspected or viral agents, in case of diarrhea, there is presentation of diarrhea. The agents include rotavirus, norovirus, astrovirus, calcivirus. They are detected by or detection of virus particles in stool specimen by electron microscopy. Again, detection of viral antigen by ELISA or detection of nucleic acid that includes RNA or DNA by PCR in stool specimen. If parasites are suspected, if there is Entomobia histolytica, its intestinal presentation is dysentery. Stool microscopy detects propozoite or quadrinucleated round cyst, and again detection of specific antigen by specific genes in stool. Now, if there is Giardia lamblia presentation fatty diarrhea, propozoites of Giardia, they are detected with 4 pairs of flagella or by tetranucleated oval cyst in central axoneme. If there is trichurus, presentation dysentery. What can be detected in stool? The barrel-shaped eggs with mucal plugs at both ends and they are bile-stained. If there is intravenous vermicularis, presentation nocturnal or pruritus, then what can be detected in stool? The plano-concave egg containing larvae which are non-bile stained. If there is suspected in Ascaris lumbricoides, commonest name roundworm, dysentery or जो भी presentation है, it's a malabsorption. Then what can be detected in stools of fertilized eggs which are round or oval, thick albumin coat, float in saturated saline, bile stained, unfertilized egg, again elongated, thin albumin coat, does not float in a saturated saline or bile salt. If there is hookworm, presentation diarrhea or anemia, what can be detected in stool? The egg can be detected in stool which contains segmented ovum with 4 blastomeres which are and the egg which is non-bile strain. If there is spondyloids, presentation diarrhea, detection of rhabditiform larva in stool by microscopy. Now treatment. Treatment depends upon the type of severity of these diarrheal diseases. First is fluid therapy, that is rehydration, is the mainstay of treatment. Then anti-motility agents and adsorbents may be considered in moderate to severe diarrhea. Then empirical therapy. Empiric antibiotic therapy is required only for severe diarrhea. Now treatment recommended. If there is mild diarrhea, what is the definition? 1 to unformed stool per day or minimal symptoms, or there is minimal symptoms or no interference with daily routine activities. Treatment recommended is hydration fluids, then lactose free diet and avoid caffeine. No antibiotic is required. If there is moderate diarrhea, that is 3 to 5 unformed stools per day with or without symptoms. Again, there is interfere with daily routine activity. Then fluids plus anti-motility agents, which includes loperamide plus adsorbents, that is bismuth salicylate, no antibiotic required. If there is severe diarrhea, that is more than 6 unformed stools per day, again there is temperature, also there is blood and fecal leukocytes in stool, then empirical treatment is recommended ciprofloxacin or levofloxacin for 3 to 5 days alternatives. Also azithromycin for 3 days for Campylobacter. Metronidazole or vancomycin for 10 to 14 days if there is Clostridium difficile. Other infective syndromes of gastrointestinal tract. First is acute nausea or vomiting, necrotizing enterocolitis, necrotizing enteritis, pseudomembranous enterocolitis, and tropical sprue. Other infective syndromes GIT continued. Sixth is esophagitis, gastritis, appendicitis, diverticulitis, typhlitis, and Whipple's disease. Okay, thank you.